Good evening, my name is Michelle Alvarado. My topic, clonal hematopoiesis and risk of atherosclerotic cardiovascular diseases. Definition: set of cells arising from a single cell are produced by a mutation of DNA in a pluripotent stem cell of the marrow or in a very early progenitor cell. Somatic mutation in hematopoietic cell pose a new risk factor for the development of cardiovascular diseases. The TET2 gene is one of the most frequently mutated in blood cells of elderly individuals. It increases the size of atherosclerotic vascular lesion. Video.
Clonal hematopoiesis of indeterminate potential, or CHIP, is defined as the expansion of hematopoietic clones carrying recurrent somatic mutations in several genes. Persons with CHIP have an increased risk of all-cause mortality and coronary heart disease. Clonal hematopoiesis increases in frequency with age. 10% of those over 70 years of age harbor these mutations. Traditional risk factors such as high cholesterol, hypertension, and type 2 diabetes may account for a large proportion of the risk for coronary heart disease. But could CHIP also be the cause of atherosclerotic cardiovascular disease? In an analysis of 4,726 participants with coronary heart disease or early onset myocardial infarction and 3,529 matched controls from 4 case control studies, investigators used whole exome sequencing to detect the presence of CHIP in peripheral blood cells in both groups. The carriers of CHIP had a risk of incident heart disease that was 1.9 times the risk in non-carriers. In an analysis of participants less than 50 years of age with early onset myocardial infarction, the risk of an MI in those with CHIP was 4 times the risk in those without CHIP. The investigators also engrafted bone marrow with loss of TET2 function into atherosclerosis-prone mice. TET2, a gene involved in regulating DNA methylation, is commonly mutated in CHIP and is associated with coronary heart disease. As compared with controls, mice with loss of TET2 function developed larger atherosclerotic lesions in the aortic root and aorta. The authors conclude that somatic mutations in hematopoietic cells contribute to the development of atherosclerosis and coronary heart disease. Full study results are available at nejm.org.
Application in nursing. Talking vital signs. Perform electrocardiogram on the patient. Medicines administration. Take blood tests. Recommend the patient to perform physical activity. Pros and cons. Advantage. This mutation provides a competitive advantage to mutant hematopoietic stem cells, allowing their progress clonal expression, which has been termed clonal hematopoiesis of undetermined potential. disadvantages. The presence of this mutation in blood cells with an increase in the cardiovascular mortality rate due to a high incidence of atherosclerotic CVD, such as myocardial infarction or stroke. Easily applied in Ecuador. Not applied in Ecuador in Boston University School of Medicine or study focus on the TET2 genes. We use misdeficient in TET2 and competitive bone marrow transplant in mice susceptible to the development of atherosclerosis. To know this clonal expansion induces a 60% increase in the size of atherosclerotic plaque in our study suggests that the somic mutation in TET2 observed in humans contribute to the accelerated development of atherosclerotic. Video.
I was asked to speak about clonal hematopoiesis. So this is a concept where cells in the bone marrow, particularly stem cells, acquire mutations that then give them some sort of selective advantage where they can grow and dominate the marrow. And we recognize that this must be a precursor event for all of the diseases that we're studying, the myeloid disorders and other types of blood cancers. And we are trying to understand how the presence of these abnormally expanded cells infer a risk of developing a subsequent disease. So my talk was focused on recognizing clonal hematopoiesis in a variety of different contexts and trying to understand what it means in those different situations and what aspects of those expanded clones might be considered adverse and which might be considered favorable. One of the things to recognize is that clonal hematopoiesis, that is, clones in the bone marrow that have acquired mutations, are remarkably common. And particularly in older individuals, we can find them in 10 to 15% of people without really having to look too hard. In other words, that these are not rare cells in the bone marrow. These actually are responsible for somewhere between 10 to 20% of all the blood cells in the marrow and the peripheral blood. So in that context, we see this age-relating association. And it turns out that those patients do have a slight increased risk of developing a blood cancer, but it's actually not overall that large. So we're trying to understand better what it is that makes some of these people progress and others not. So in other parts of the talk, I also discussed other contexts where we see clonal hematopoiesis. So for example, patients who have aplastic anemia, which is a disorder characterized by an immune response against blood cells. We see that almost all patients, particularly older patients, have small mutant clones that are present in this disorder. And some of the clones tend to be more adverse than others. Some of them are trying simply to avoid the immune system, and therefore they're trying to mutate in a way that provides an immunologic escape. Other mutations are more like the mutations that we see in MDS and AML, suggesting that these clones have found a different way to survive, maybe by proliferating better or being more resistant to apoptosis. And we know that those patients are the ones who actually are at increased risk of developing a subsequent problem or of simply dying from their aplastic anemia. I think in particular the people that we're going to run into clonal hematopoiesis most often clinically are people who have low blood counts to begin with, people that we suspect might have a blood disorder. So if they don't meet diagnostic criteria based on what we consider disease states today, we can sequence these people and identify that about 40% have clonal hematopoiesis. And we know that these patients are at increased risk of developing a subsequent malignancy. So we're therefore able to identify those patients that we need to worry about more, the ones we might need to follow up more often, and identify those patients that don't have mutations and are probably at lower risk, where we might look for an alternative cause of their low blood counts, or at least reassure them that they're unlikely to develop problems in the near future. So it can change clinical practice in terms of how we predict what our patients are going to be like. But I think in the future, we might actually have some way of intervening, that if we know that patients that are at risk of progression, we may have mechanisms to try to prevent that progression. And this allows us to at least identify the group of patients that we should focus our efforts on.
In conclusion, in my opinion, mutant hematopoietic cells have been shown to be associated with a high incidence of atherosclerosis, but it is being studied that LAB block or inhibition of inflammatory NRPP3 inhibitor or this system are the subject of the Canton's clinical trial for the treatment of ECF. Bibliography. Thank you.